So, hi everyone, can you hear me? Perfect. So, my name is Panagiotis Lalagkas, and today I will be talking to you about one of my projects during my PhD, in which we leverage shared genetics between breast cancer and its predisposing diseases in order to identify candidate drugs for repurposing for breast cancer. So, the big goal of my PhD overall is to discover novel effects of existing drugs by using genetics data. And in one of my earlier works, I analyzed pairs of Mendelian and complex diseases, in which pairs the Mendelian disease, acting as a risk factor, increases the risk of the complex disease. And this is happening because of the pleiotropic effects of the Mendelian genes, which on the one hand, they cause the Mendelian disease, and on the other hand, they increase the risk of the complex disease. So for this reason, we can say that these genes, these Mendelian genes, are actually shared between the two diseases. So the idea here was that if we find drugs that target these shared genetics, which in this case are the Mendelian genes, then we might have drugs that can be useful for the treatment of the complex disease. And this is the work I presented last year at ISMB in Lyon, in which we showed that these drugs are actually enriched, significantly enriched for drugs already investigated or indicated for the complex disease. So after showing that, we want to go a step further and ask, what if we now analyze pairs of complex diseases? So right now here we have a complex disease being as a risk factor that increases the risk of another complex disease. And again, the idea here is similar, it's the same. If we can somehow identify the shared genetics between these two complex diseases, then we can find drugs that target those genes and then propose them as recommended candidate drugs for repurposing for the complex disease. But here, because the risk factor is a complex disease itself, we can focus on the drugs that are already known to treat that risk factor and try to identify a subset of them that target these shared genetics. So the hypothesis we make here is that genes that are shared between a complex disease and its risk factor can suggest new uses for drugs known to treat the risk factor. And to evaluate our hypothesis, we used breast cancer as a target disease because it is a well-studied disease and also it has known risk factors. And in fact, we found in the literature six other complex diseases that are breast cancer risk factors. So let's see how we test our hypothesis. So as I said earlier, the first step is to identify the shared genetics between the two diseases. And the way we do it is by using GWAS summary statistics data for both of them, and a tool that's called LogoDetect, which essentially does local genetic correlation analysis. So this means that this tool, LogoDetect, analyzes both GWAS of both diseases at the same time and tries to find genomic loci that include SNPs with correlated effects in both diseases. So for example, in this mirror Manhattan plot, you can see at the top, this is the GWAS for breast cancer. At the bottom is the GWAS for, I think this is high cholesterol, for example, which is one of the risk factors for breast cancer. And the red dots are the SNPs within loci that the tool found that includes SNPs with correlated effects between the two diseases. So after identifying these shared genetics, we have to map them to genes. And there are many different ways we can do it, but in this project, we chose the most simple way, which is actually we focused on the positively correlated genomic loci, and we got the SNPs, the genes, positionally located with this locus. So in this example here, if we say that the pink area is a positively correlated genomic locus, gene A and gene B will be part of the shared genes between breast cancer and this risk factor, but gene C will not be. However, because we start from loci with correlated effects, we don't expect all the genes positionally located in these loci to actually participate in common biological processes between the two diseases. So in order to prioritize the more likely shared genes, we use two other tools, and one of them is MAGMA, which aggregates the effect of SNPs within a gene on disease risk, and the other one is the S-multi-X-scan, which associates SNPs impacting gene regulation, the EQTLs, with disease risk. And using these two tools, we prioritize the genes that are significantly associated, at least with the risk factor, and also they are genetically predicted to be dysregulated towards the same direction. And in order to make it more clear, I have this visualization here, so you can see that these are the 7 genes we identified to be positionally located within the positively correlated loci between breast cancer and high cholesterol. And all of them, as you can see by the color, found to be associated by magma with the risk factor, with high cholesterol. However, after using the multi-X scan, we found that only 3 of them, the ones in the green area, to be predicted to be dysregulated towards the same direction. And these 3 are the subset of genes we say that they are part of the shared genes between the 2 diseases. So now that we have a list of shared genes, the next and the final step is to find which drugs that are among the ones already approved for the risk factor actually target those genes. And the way we make this connection is by using canonical pathways from the MSC database. So here we use a protein-protein interaction network and a propagation algorithm to first connect the shared genes to the canonical pathways in order to identify pathological processes that are shared between breast cancer and each risk factor. And then we use the same approach to find which of the drugs already approved for the risk factor target these shared pathological processes. So for simplicity here, in this slide, I just put only one shared gene, which is the one we found between breast cancer and high cholesterol, the MLXIPL. And as you can see, we could connect it to the FOXA2 pathway. And in this case, the FOXA2 pathway is known from the literature to be involved in breast cancer pathogenesis and also lipid metabolism, which gives us confidence that our approach identifies known biology. And also, we could connect the phenofibrate and rosuvastatin to this pathway, which means that we recommend these drugs for repurposing for breast cancer, and we can also provide specific pathways and genes that might actually explain the therapeutic effect of these drugs on breast cancer. But of course, this is only one example that I chose to present to you, and we'll have to systematically evaluate our list of candidate drugs for repurposing for breast cancer. And the way we do this is by using clinical trials data. So on the right here, you can see the number of drugs that have been already investigated or indicated for breast cancer across the different drug development phases. And on the left, you can see the number of drugs that are already approved for the risk factors we tested in our case. And on the bottom, you can see that 16 of these approved drugs have already been investigated or indicated for breast cancer. And our method could identify 15 of them, which results in significant enrichment of our list of the list of our candidate drugs for drugs already known to be investigated or indicated for breast cancer, which gives us more confidence about our recommendations. So in conclusion, our approach identifies drug repurposing candidate drugs for breast cancer by using shared genetics with its known risk factors, and it also provides biological support for its recommendation by pointing to specific pathways and genes that might explain the therapeutic effect. And we find that our candidate drugs are enriched for those currently investigated or indicated for breast cancer. So if you want to learn more about our methods, and if you want to get access to the full list of shared genes, pathways, and drugs connections we make, you can scan this QR code, which will lead you to the preprint. And thank you for your attention, and I'm happy to answer any questions you may have.
Thank you.